Now, we now want to note the different categories of tonicity. Tonicity has to do with the concentration of solution compared to the solution that is on the other side of the membrane bounding it. For example, if this is a cell with its nucleus and cytoplasm, and this cell has a surrounding, if the inside of the cell is having weak solution and the surrounding of the cell has strong solution. The meaning of these two is that the weak solution inside of the cell means that the inside of the cell has more water molecules, okay, and there is less solute concentration inside than the outside. If the outside is a strong solution, it means that there is more solute, molecule. So here we have more solute molecules. Here we have fewer solute molecules. So if this is a cell, maybe this is a living cell, and this is the surrounding of the cell or the cell environment. If the inside of the cell is weak and the medium surrounding the cell is strong, we expect that water molecules will move from the weak content, from the weak solution inside of the cell to the outside. So when water molecules move from the inside of the cell to the outside, we term it exosmosis. And if it's in a situation where water molecules move from outside of the cell to the inside, we term it endosmosis. And that's not really where we are going for now. Where we are going is to mention that whenever a solution is weak and is being separated from another solution that is very strong, we say that solution that is weak is hypotonic. So hypotonic solutions are actually weak solutions. And the solution that is strong is said to be hypertonic. So this solution that is strong is hypertonic to the content of the cell. And the solution inside that is weak is said to be hypotonic to the environment of the cell. So in short term, hypotonic solutions are weak solutions, whereas hypertonic solutions are strong solutions. They are strong solutions. Now the third variation of tonicity is the isotonic solution. The isotonic solution. Isotonic solutions are solutions that are at equilibrium. So for tonicity, we have three types of solution. One, we have hypotonic solutions, which are otherwise called what? Weak solutions. Then two, we have hypertonic solutions, which are what? Strong solutions. And three, we have isotonic solutions. which are solutions at equilibrium. These are solutions at equilibrium. That is solutions of equal concentration. Solutions of equal solid concentration that are separated by a semi-permeable membrane. So they are said to be isotonic to one another. You understand it now? Now, if a solution is strong, that solution is said to be hypertonic to another solution or another medium that is weak. If a solution is weak, that solution is said to be hypotonic to another solution on the other side of the membrane that could be as well, that could be a very strong solution. And so that means that between two sides of the membrane, there could be concentration gradient. Difference in concentration is what is called the concentration gradient. And the concentration gradient gives rise to what we call osmotic pressure. Gives rise to osmotic pressure that exists between the cell and its environment. Naturally, living cells tend to maintain isotonic condition for normal functioning. Living cells, they tend towards maintaining isotonic condition. And that is why if between a particular cell and its environment there is a concentration gradient, or if there is osmotic pressure being developed, then there must be osmosis to bring about a balance. Most cells will do well when there is an isotonic condition. So all cells will tend to allow for movement that will create equal concentration of water molecules within and without. And that's why this osmosis is very important in the body of living organisms. Cells tend to maintain isotonic conditions. Now, what gives rise to that is that if the surrounding of a cell is concentrated, if this is concentrated, and the inside of the cell is weak, then there is going to be high osmotic pressure. Osmotic pressure will be developed on this outside. And when osmotic pressure is developed here, then water molecules will tend to move out of the cell. Why? Because the solute concentration is high here, the solute concentration is low here, because it's a weak, it's a very weak, it's a weak solution. So that's that about the types of solution and tonicity and so on. Beside that, we want to move forward to look at the conditions that are necessary for osmosis to take place, especially in plants and animals. Conditions necessary